let's look at a way of looking at organisms and seeing how they move. Okay. Um, you can see the lovely little guy. Here's a cover slip, and they get all smashed. Okay. So first of all, motility is the movement of a bacteria. Some bacteria are motile. Some are more motile than others. Can spread over an entire agar plate. But basically, you want to know whether these guys are motile, and usually they're by species. And this becomes important because if you have a motile organism and it gets into a certain area, it can move. All right, from one spot to another. So, like you get some of these proteases and UTIs and stuff like that, they're very motile. They'll make it up the entire urinary tract. Okay, so you do want to be aware whether an organism is motile or not, or at least the physician needs to know. So we want to look at this, and there are different ways that bacteria move to a more favorable environment or away from unfavorable environments, and they can do it in several different ways. You can see this chemotaxis, they are attracted by chemicals, a particular chemical or nutrient they need. Phototaxis, they could be attracted by light, or aerotaxis, attracted by higher oxygen content. Now, how do they move? They move by generally flagella. I mean, some of your protozoans do something different, but for bacteria, they move usually by their lovely little flagella. And we use the word trichus. Trichus meaning hair. That's what it translates to. So monotrichus, one flagella. Amphitrichus, and I'll show you what they look like. A flagella at each end of the cell. Peritrichus, a flagellum on all sides. Think of a periscope. And Lophotrichus, several flagella on one side. So when you look at this is the monotrichus, this is the amphitrichus, this is Lophotrichus, this is the peritrichus. Okay. All right. So the other thing that we look at is when we put these, we put a drop of water with this little organism. Basically, you can pick up the organism, put a drop of water on the slide, mix it up, and look at this. You got to be pretty quick under the microscope because it does dry. But there's several things that we're looking for when we put this underneath the microscope. You can see three different kinds of movement. One is called Brownian movement, and this is when you put this drop with the bacteria in there under the slide. There is random bombardment of molecules due to the water that's going on. It's not true movement, and what you see underneath the microscope, unfortunately I can't show you, is these organisms shake or jiggle. They're in one place and they're shaking. And again, we call that Brownian movement. A lot of times when you put the water in the slide, as it heats up, the water moves a little bit, and it kind of sloshes. And that also is not true movement. The bacteria will kind of go with the flow. They'll move from side to side or wherever the water is moving. The way you decide that these organisms are truly motile is you need to see them change direction. They will rapidly swim. They will abruptly make a 45-degree angle and take off somewhere else, and they may swim against the current. So that's what we're looking for. And so when I'm in a microbiology lab and I want to show this motility, that's what we're looking for. And it's like, ignore the going with the flow, ignoring the jiggle, just go for these changes in direction. Okay. And this is how you make a wet mount. Okay. You take the organism, you add a couple drops of broth or water, and you take that cover slip and you drop it right over them, and they get squished a little bit. Okay, and that's what we want to see. That's how we determine whether an organism is actually motile or not. That's how, through research, we found out how these guys were. And then we went and looked for their flagella, and that's how we determined the type of flagella. So, I mean, some of these swim very well in one direction, some of them are all over the place. Well, sometimes trying to make a wet mount, it's a little difficult. But another way you can do this is you use sim auger, sulfide indole motility auger. And this auger is semi-soft. It's softer than regular auger. It doesn't gel as much, and it allows the bacteria to move through the medium. And so what you're going to do is you stab the medium. I'll just show you, so the medium would be there. You take this needle and you stab it. And any non-motile organism will only grow along that stab line or that inoculation line. Motile bacteria will grow along it and away from this. And I'm going to show you this in a second. Okay. So what you do is you flame your needle, you dip the sterile needle into whatever organism you get, you stab the sim auger halfway down one time. Don't wiggle it. It is like a real quick down and out the same way. This is sometimes hard. Most of my students get a kind of V shape. And then you inoculate it at 37 degrees body temperature for 24 hours. And this is what you get, okay? A negative test is this straight stab down, whereas positive, you can see that organism grew away from that initial stab. So that's a very easy way to determine. And that's it for the motility test, okay?